Today, I will be talking about the paper titled Single Cell Lineage Analysis Reveals Genetic and Epigenetic Interplay in Glioblastoma Drug Resistance. The bioinformatics method is sequence filtering. First, introduction. The scientific question is, can certain genomic amplifications of loci in glioblastomas help find the cancer's resistance to drug therapies? The hypothesis is, if a combined lineage tracing / barcoding and single cell RNA sequencing is treated with receptor tyrosine kinase inhibitors, then it can reveal the interplay of genetic and epigenetic mechanisms of drug resistance in longer term dasatinib resistant cell populations. Next, we have the key terms of the paper defined. 1. Single cell RNA sequencing. This is a method of sequencing the same tissue type to find different functions and jobs of the different cells present. 2. Dasatinib resistant cell populations. These are cancer cells that have a resistance to tyrosine kinase specific inhibitors. 3. Genetic and epigenetic mechanisms. These are acquired genetic events of persistent phenotypes. 4. Malignant cells. These are cancer cells that grow and spread throughout the body by metastasis. 5. We have the receptor tyrosine kinase inhibitors. These block the tyrosine kinases, which are usually high in levels in cancers, to keep cells from growing. 6. We have GSC8 gliomospheres. This has the PDGFRA amplification that allows it to adopt a drug-resistant phenotype to survive initial drug treatments. 7. We have the TSNE visualization. These reveal a strong coordinates between the two experiments that will be talked about later on. And this also reveals two main clusters of cells that are distinguishable by the expression of cell cycle genes. Next are the take-home findings. First, the scientists combined a lentiviral transgene barcoding system with a single-cell RNA sequence. Here, the DNA barcodes obtained would be transcribed and then captured in single-cell RNA sequence. With the diverse barcodes being transcribed, rare subclone/tumor cells would be detected in the malignant cell populations in their DNA sequencing analysis. Next, the scientists performed two experiments. One experiment will be used to target DNA sequencing and assess barcode distributions. Then the same experiment was performed, but with a separate parental culture of GSC gliomospheres with cellular lineage barcodes for the second experiment. The first experiment had dominant jackpot lineages that made up 18.2 to 65.5% of the cell population replicants that represented more than 2,000-fold expansion of cells after just 70 days. The second experiment had all 6 replicates containing a jackpot lineage between 52.4 to 88.5% of cell population replicants in just 56 days, which were already visible around 28 days. This meant that it must have an early advantage to resist the drug treatment. The second experiment was also dominated by the same lineage being the most abundant in the same barcode ID, E86 back. Experiment 2 displayed how genes in the E86 back lineage have many genes residing in a single chromosomal band, chromosome 13. The role of this chromosome amplification in RTK inhibitor resistance have been reported in specific cancers. The transcriptional programs of these individual cells were analyzed by individual cells for the TSNE visualization. This suggested that cell populations from the same patient-derived gliomospheres are adapting to targeted RTK therapy. Experiment 1 replicatively displayed some degree of drug-resistant reversibility in clonal isolates with a high copy number amplifications that had more stable dasatinib resistance. This concluded that non-jacked-up genes lost their drug-tolerant phenotype when cultured in the absence of dasatinib consistent with reversible genetic resistance mechanisms. This concluded that combined lineage tracing and single-cell RNA sequence revealed that there was an interplay between complementary genetic and epigenetic mechanisms of resistance and heterogeneous glioblastoma. Why is sequence filtering important? First, it helps you find similar sequences that will be inserted in the database. Second, it can help isolate certain parts of the sequences, like you can see on the right. Third, it can look over the entire database for comparison, which will help you find rare sequences you might not have heard of, especially for cancers, like what they are doing in this paper. Next, why is this important and what has been done before? The paper talked about how low coverage whole genome sequencing was used previously. This was like the next generation sequencing. It was used in clinical oncology to advance the treatment of cancer. It was also used to identify rare cancer mutations by sequencing large number of genes in a single test. The most recent publication that came before this study was done by Tangina Cater in 2016. There, she used low coverage whole genome sequencing in embedded tumor tissues. How are the results presented different? In this study, they focused on single cell analysis rather than a large number of genes. A mixture of single cell RNA sequence and lineage tracing were used to find the interplay of genetic and epigenetics and the resistance of the tumor cells to drug therapies. This study was able to isolate a specific chromosome that related to the drug resistance. What are the limits of this study's applicability? The limitations of this study that make it harder to reveal the interplay of genetic and epigenetic mechanisms of drug resistance come from how they performed the experiments and how they interpreted the results from the reads. The reads that they sequenced were very lineage and chromosome specific. Because of the technology they used, low expression genes were hard to detect. This was not taken into account when they concluded their study. How was bioinformatics method used to support the results? To the left, you can see that figure 2 displays how single-cell analysis helped identify the chromosomal amplifications and jackpot drug-resistant lineages. Before going into depth what figure 2 is, I want to ask, what is sequence filtering? Sequence filtering is when you isolate certain tissue cells and focus your sequence on that same tissue cell. Sequence filtering uses specific programs to be able to identify particular types of sequences or find rare types of sequences. This narrows down where the cancer cells are most abundant, such as in specific chromosomes/lineage. The scientists were able to find early cell growth replicates that led them to suggest that many cancer cells were found in a singular lineage. They resided mostly in a chromosome. I will now be shifting my focus to figure 2a. This figure is showing a plot depicting barcode distributions in single-cell RNA sequencing data for the 1,268 individual cells that were from experiments 1 and 2. If you look towards the left axis, you see these blue circles. These blue circles indicate the number of barcodes that were detected in the number of cells, which is the x-axis here. The red line on the right axis indicates the cumulative fraction of cells for which the linear barcode was detected. The take-home message of this figure is this E86 back. The E86 back shown here is considered the jackpot lineage, and this was identified through DNA analysis of experiment 2. It had the largest number of cells detected at 30 single cells. This is important since it appeared the most in the lineages and helped the scientists narrow down the lineage of where the drug-resistant cancer cells were coming from. Here you have an example of a BioPython code filtering a sequence file. This first line of code is where you import the biosequence. These next lines of code are examples of simple text files. This first input file is a function to read in the gene expression file. The ID file has all the IDs as FASTA genes. This output file is where you output a FASTA file with one or more of the sequences. Here you have the with statement. Though this part of the code is a little more advanced, the wanted line here is a variable that pulls the IDs that you want. Then you are printing out the unique IDs in the ID file. For records, this line of code is where you're parsing the sequences and the records. For count, this is returning the amount of times the sequences you parsed have occurred in the files. Then you are printing out the saved records. Finally, you are printing out the IDs not found in the files. There are some questions I have about the code. One, is there any way this code can narrow down a certain sequence all the way down to a certain chromosome? Two, how can two different data be pre-processed in this sequence filtering all at once? Three, how big of a sequence can be allowed to be processed in this code? Is there a limit?